Hello, everyone. It's a pleasure to be here and present the work that we do in the lab. And for today's presentation, I'm going to present where we're comparing in vivo and in vitro tissues using single-cell genomics.
Gracias, Rosé. Perdona que tenía un problema de conexión. Voy a presentarte primero. Hago la presentación en español. Como habéis visto, la charla va a ser en inglés. Las preguntas podéis elegir en español o en inglés, como queráis. Pues os cuento sobre Rosé Vento y su grupo. Ella se centra en comprender la influencia de los ambientes celulares y las respuestas en células individuales, sobre todo en el contexto del desarrollo y de la inmunidad. Su equipo emplea métodos de transcriptómica espacial de células únicos para deconstruir las señales celulares, sobre todo en órganos y tejidos humanos, y utiliza toda esta información para trabajar con modelos in vitro. Su equipo desarrolla herramientas para construir redes de interacciones entre células a partir de estos datos transcriptómicos. En su grupo han desarrollado herramientas como cellphonedb, que es una herramienta para estudiar la comunicación célula-célula, y esta se ha utilizado, por ejemplo, para desentrañar la comunicación entre células maternas y células fetales en lo que sería la interfaz entre útero y placenta, en lo que serían las primeras fases del embarazo humano. Rosé tiene innumerables publicaciones y proyectos, algunos financiados por agencias internacionales como Horizonte 2020, Marie Curie, Wellcome Lib, y de hecho recientemente ha recibido el premio Early Career Research Award de la Biochemistry Society. Y no me entretengo mucho más. Rosé, muchas gracias por aceptar la invitación y cuando quieras es todo tuyo.
Hello, thank you. Thank you for the nice presentation. It's a pleasure to be here and present the work that we do in the lab. So for today's presentation, I will be focusing on our work mapping tissues in vivo and in vitro using single-cell genomics technologies with the idea to benchmark in vitro models and also improve them. So in a more general level, what we are interested in the lab is in understanding the principles of gene regulatory circuits with a specific focus on how the tissue microenvironment influences them. So what we want to understand is how cells develop and acquire their identity, start expressing specific genes, and how are those really regulated by the gene circuits, and really how they then start self-assembling and creating more organized tissues. But also how this process, which really is not linear, and how then the adjacent cells in the tissue fit back into these gene circuits, and this again influences the identity of a cell. So to decode these relationships between the microenvironment and the gene regulatory circuits, what we do is to use single-cell transcriptomics or single-cell genomics tissues in different biological systems, and we have a particular focus on the immune system and the reproductive tissues. So what we do is to integrate multiple layers of regulation. So from single-cell transcriptomics, where we can group cells based on similarities on their transcriptome, and then that gives us information about the identity of the cells and potential functions. We also use special transcriptomics, which allow us to assign specific cellular coordinates to this cell state, and this is important to decode the cell architecture or the tissue architecture, and also start understanding how cells that are close to each other talk to them. So really constructing cell-cell communication networks. And finally, we also look at other layers of genome regulation by using, for example, single-cell ataxic. And what this really allows us is to understand how these gene circuits are encoded. So key for really integrating all these multiple layers is the bioinformatics and machine learning tools that we use. So what these bioinformatic tools allow us is to really query these integrated data sets by, for example, asking what are the cell states that are active or present in a specific data set. Also, what are the gene programs that really inform us about the potential function of a cell. Also, with the spatial transcriptomics, we can ask with the computational tools what is the location of a cell, and also the cell-cell communication networks. So a lot of you may be familiar with single-cell transcriptomics. That's why I will not go into much detail for this. But I will spend 2 slides talking about the spatial transcriptomics, and in particular the Visium technology, that is the one that I will be using through my presentation. So in this technology, we have the gene expression slides, where then we put the tissue on top of these slides. And by difference to normal tissue slides from histology, these slides contain some capture areas with barcoded spots, which basically these barcoded spots contains also a special barcoded capture oligonucleotide, which means that when the RNA is hybridized here, you also have information about the specific space of where this RNA is coming from. Which means that then we will have, in addition to the HNE, so the structure of the tissue, we will have information about the expression of this transcriptome in this spot of around 50 microns, which contains about 1 to 25 cells. And then really, because this is not single cell resolution, we need computational tools to make sense of the data. So for example, what we use to understand the identity of cells in these spots is cell-to location, which is a probabilistic method that basically decomposes these spots based on the transcriptome from that single-cell transcriptomics data. And what really this gives us is an approximate information about the cell states that are present and the proportion of those cell states in this particular spot. So in this way, we have a calculated probability of the cell states that are present in the different regions or niches of the tissue. In addition, we are interested in how cells talk to each other, and for this, we use cellphonedb, which I developed when I was a postdoc, and then my team has bring it to the next stage by considering not only communication through ligands and receptors, but also considering cells that coexist in a specific space. So let me talk a bit more about cellphonedb. So basically in cellphonedb, it contains on one hand about a database, and on the other hand also has a method. So in respect to the database, we have cellphonedb, which basically contains information about the interacting ligands and receptors. What is unique is that we consider multimers, which means that one receptor is can be formed by multiple components, which in the gene expression means that you need more than one gene to be expressed in order for this receptor to be functional. And on top of that, we have another database now that is called CellSign, in which we have annotated receptors and transcription factors that are part of the same pathway. Okay, so on top of the database, we have the method. And now, before we were doing random shuffling, but now we have simplified, and what we do is to look for ligand receptor expression by cell using single cell transcriptomics, and we do differential expression between the conditions that we want to pick in our data. So then we filter those interactions, as I was mentioning before, by getting information about the cells that are coexisting in space. And for this, we use special transcriptomics. And for example, by coexisting, what we can use is just histological information to define the niches, but also special clustering or NMF. And finally, we look for active downstream signals. So then this way we can say, okay, this receptor is likely to be functional because the downstream response is active. And for this, what we use is single cell tag, because this way we can calculate transcription factors that are active in a cell, and this is what we use as the sensor of the activity of the receptor. Okay, so with all of this information, we are generating atlases. So basically, what we do is with the single cell transcriptomics, we define cellular identities, but then the spatial transcriptomics together with the computational methods that I was telling you allow us to look into spatial atlases and really have a more assembled of a coherent version, a holistic version of the tissues. And this allows, in a way, to read the different organs that we profiled. And in particular, we have been doing atlases of multiple organs, including the uterus, the gonads, and also the placenta. So one of the things that are most excited about this, about the fact of having these dense atlases of the human tissues, is actually start doing in vitro modeling, so not going from reading organs to start writing them, to start creating them in vitro. So for example, from the single cell transcriptomics atlases, we have the information of the different identities of the cell state, but also the regulatory pathways that are driving the differentiation. And that really we can use as a template to then induce differentiation by adapting the media in culture. Likewise, all the spatial information can then be used to start putting these entities, isolated entities that we create in a dish, together to make a more complex systems, such as some co-cultures with hydrogels, and also even organ-level systems like organs in a chick. And this is something that we have been also exploring in different organs, including iPS macrophages, and we have been successfully really using this information to promote one lineage or the other in case of the in vitro. So for today's presentation, I will be focusing or giving an example on how we mix this in vivo and in vitro data. In our case, in our recent manuscript of the human placenta. Okay, so let me give a bit of introduction of the human placenta and why this was a really important question for us to understand, and where the single-cell genomics and the bioinformatics play a crucial role in the project. So basically, what we are interested is in early placentation. and in particular in this communication between the fetal placenta and the maternal uterus. So this is a unique window or development where really two organs from two different individuals, mother and fetus, coexist together to have a coherent function. And really what is fascinating about this process is how this mother and fetus communicate. So what happens is that we have the trophoblast, which basically are the epithelial cells from the placenta with the touch from the placenta and start invading the uterus. So as they invade, these trophoblasts acquire an invasive phenotype, and what they will do is to destroy the arterial medium. On the other hand, they also go inside the arteries and then acquire a vascular phenotype. And really this again helps with this transformation of the arteries. And the reason you need the transformation of the arteries is that then you have maternal flow going here, and this enables the exchange of the oxygen and nutrients between mother and fetus. So our questions for this is what are the molecular mechanisms that trigger and stop this invasion? And this really is because both that these trophoblast cells do not invade or that invade too much is problematic and are associated with pregnancy complications. And the other question is how these trophoblasts acquire this invasion phenotype or this vascular phenotype, and what are the regulatory mechanisms that drive this process. And the reason we are excited is because if we can resolve this question, then we can start understanding how successful placentation works. So the limitation of this is that these highly invasive trophoblasts are actually not present in mouse models. So that really means that the best model to study is the humans, which then comes with limitation because you need to profile early pregnancy. And also, the problem is that this deep invasion is not present in human accessible samples. So when I published my postdoctoral work in Nature in 2018, I was only profiling this early invasive phenotype, but not the last one that will transform the arteries. So one of the questions that I asked myself when I started my group is, how can we look at the stages of the trophoblast invasion? So really the only way to look at this is to interrogate these samples that are from the first trimester hysterectomies, uterine implantation sites, and those are not collected nowadays. There are, we have a biobank here that is collected by Professor Ashley Moffett, where they have stored some of these samples that are frozen. So really, how can we make use of these historical collections to start understanding this invasion? And this is where genomics and bioinformatics came from. So before I go into detail for the analysis, let me show you how fascinating this process is. So here we have the placenta, and here the decidua, which basically is the uterine tissue. You can appreciate how different the histology is. And if we zoom in, here you can see the trophoblasts start getting into the uterus and invading. You can also see, as I told you before, how these arteries get transformed, and you can see these vascular EVTs, which have this big shape invading the fetal tissues. Also, in the end, they fuse, and then you have this big multinucleated cell. And then you have also this small dot, which basically are maternal immune cells that coexist and give signals to the trophoblast. But what we want to know is what are these signals and how this process of invasion is happening. Okay, so what we did here was to take the tissue blocks and then profile using single nuclei. And what we did on this single nuclei was to then characterize the cell states. And then here, while there is a lot of names that you don't need to remember, what is important is that you can see how this is the trophoblast, different states of the trophoblast. And you can see the amount of heterogeneity that we see by the transcriptome that you apparently don't see or was not characterized into the tissue blocks. And what is also more interesting is that then if we reconstruct trajectories, so the time trajectories based on similarities on the transcriptome, basically you organize the cells based on how similar they are on the transcriptome. And then you can define here our progenitor states and how they start invading, which is here towards the uterus on the left, and how they split into these two pathways that I mentioned before, and that was not was only known because of the histology, but now we can start digging into the molecular programs that gives this invasion. But before we go into the invasion, we want to have more insights about this trajectory and also about the different identities that here we just put based on inferences on the transcriptome. And here is where we use the spatial transcriptomics with Visium, where we deconvolve the spots based on probabilities of the single-cell transcriptomics. And clearly you can see what I was telling you before in one image, where here on the left, you have the trophoblast that will go to the vascular phenotype, and you can see here forming a plaque and the probability, while you can see on the right how the trophoblasts invade, and you can see the different colors really matching and really validating what we see in the single-cell transcriptomics data. So it's really the back and forth between the annotation from the transcriptome with the spatial data, what really give us the identity of those cells. Here I'm just showing one image, but in the manuscript you can see how we did this with different colors. So then we also wanted to organize cells not only based on single-cell transcriptomics, but also adding multiple layers. And here we decided to use the spatial data because as they invade, they differentiate. So in this case, the pseudo space really matches the pseudo time. So what we did is, together with Oliver Stigall, to design this ST order, which basically is a statistical method to order cells in space. And basically, ST order takes as an input the estimated cell identities in Visium. Then it feeds a Gaussian process model that derives pairwise spatial covariance scores for all the cell state pairs. And what this gives us is an organized trajectory, which it was very comforting to see that in our case really matches what we see on the single-cell transcriptomics, again confirming how in this case pseudo-time and pseudo-space are the same. Okay, good. So we have a trajectory, and the reason why this is important for us is that then we can start understanding what are the regulatory programs that mediate this transition, so cells differentiate what identity they acquire. So in this case, what we did to calculate the transcription factors were to get a consensus from multiple measurements. First was that the transcription factor is expressed and changed its expression. The second one that is active, and we measured active by two ways. First was using the consensus target of specific transcription factors, and for this we used Dorothea. So if the consensus targets are significantly upregulated, we consider the transcription factor as active. And the other way was to look at the open chromatin and looking if the binding sites for the transcription factors are active. And with all of this, we have a consensus. And then we did a model of how this differentiation really how is induced by changes on the transcription factors, and really matching the regulome with the specific cell identities. And what was really good also for us to validate is that we have described previously that in the uterus there are gradients, signaling gradients, so as for example wind and notch, and we can really now match how these gradients are affecting the invasion of the trophoblast, really validating these or making a holistic model of this process. Okay, so then we got into the fun where we really wanted to ask how this maternal and fetal communication in the uterus work. And there are two questions that we asked. First is how this vascular transformation happens. And the second one is what drives the invasion. And for today's presentation and on interest of time, I am just going to focus on the cell-cell communication events that drive trophoblast invasion. Okay, so what we did here was to use cellphonedb, which, as I mentioned it before, looks at the ligands and receptors of cells that coexist in space. And we look interactions between the maternal immune cells and these invasive trophoblasts. And then that gives us a model of multiple chemokines and cytokines that can be influencing this invasion. So the next step was to really validate that those cells really are close to each other and the ligand receptors expressed are also close. And for this, we use a better resolve spatial technology that is called single molecule FISH. And you can clearly see how in green are the maternal immune cells, while in blue they are these big cells that are placental cells. And what we can see is how these maternal immune cells are secreting molecules and how the receptors for those are expressed by the trophoblast. And we did this on multiple immune cells, including the NK cells and also the macrophages. And really, this is a predictive model on how the immune cells may be helping with this invasion of the trophoblast. But really, to validate all of this, we needed to move into the organoids. But obviously, we have another challenge, which is to characterize what these organoids are doing. So the organoids basically are cell cultures. So as you can see here, they are placentas that can be simplified, placentas in a way that can be grown in a dish. And that allows us to perturb and really having something that can be unexpanded in vitro. And as you can see, there are two models, which I won't go into detail, the organoids and the 2D system. And basically, in both cases, what is very cool is that they can also invade, as you can appreciate here. They have this really shape that indicates that they are invading. And what we did here first was to understand if this process is the same that happens in vivo, so then we can use it to validate the data. And for this, again, what we did was to use single-cell transcriptomics. And then what we did is to utilize the information from the in vivo data to benchmark the model. And to do that, what we did is to first look at the markers that are expressed in vivo and check if they are also expressed in vitro, but also doing more sophisticated analysis where we train a model, a logistic regression model, with the in vivo data, and then we project the data to the in vitro data sets. And in this way, we can assign the different populations that we defined in vivo into the in vitro, and more importantly, also get probability confidence on how well the cell states from the in vivo are recapitulated in vitro. And without going into much of detail, here are the invasive populations. I'm highlighting them in my mouse. And what we can see is that they are well recapitulated. These early stages of invasion are well recapitulated in a dish. This is not the same for the late invasive stage, which they are not, but the early invasive one, what promotes invasion, is well recapitulated. So then what we did is to use these in vitro models to add the cytokines we predicted to be promoting invasion in vivo into the dish. And what we could find really is that, yeah, these in the presence of these cytokines, these trophoblasts start expressing motility genes, really demonstrating how this crosstalk between mother and fetus is essential to promote invasion and to promote early placentation. So to finalize my talk within the 30 minutes that I have, what we have been using is to use genomics and computational tools to leverage historical samples of early placentation to understand a process that was very unknown, and then utilize this information to understand what is the heterogeneity of cells that we are having in the dish. And what we have done is to look for and make a model of the pathways that are active as cells differentiate and start invading into the tissue. And then what also we have done is to then move into the spatial and cellphonedb to understand the communication between the maternal and fetal cells. And we have seen how maternal immune cells, and in particular, we focus on macrophages, secrete molecules that help invading the trophoblast, and we have demonstrated that using the organoids. So what we are excited about this study is first that is allowing us for the fundamental understanding of pregnancy complications, because as I mentioned at the beginning of my talk, the majority of pregnancy complications are caused by a lack of invasion of this trophoblast or too much invasion. And the second area that we are very interested on exploring is on designing the blueprint, or using this information as a blueprint for designing better in vitro models. So basically, up to now, we have characterized what do we have in a dish by just comparing to the in vivo. And as I mentioned, we have this invasion, but only the early invasion, which was okay for us to validate. But then what we want to do now is to start generating these late invasive states and cellular states with the vasculature and these few cells that currently we don't have in the in vitro model. But now we know what are the pathways, what are the regulatory networks and the interactions that we need to supply in vitro in order to have these better models and improved models in a dish. And with all of this, I will just want to acknowledge the members of my team who led this project. This was a highly collaborative work between Anna Jutnanan, Kevin Trolle, and Frederick Wong from my team, and also collaborators from Cambridge and the Sanger Institute. So thank you very much for all your attention, and looking forward to hear about your questions. Thank you.
Muchas gracias, Rosé. Es impresionante todo lo que has contado. Te doy la enhorabuena por todo el trabajo. Yo, de hecho, recuerdo cuando leí el artículo de cellphonedb, que ya me parece un poco futuro, todo lo de los ligandos y demás. Y claro, yo el uso que le veía era muy relacionado a todo lo que has estado contando, pero la verdad es que creo que lo que estáis haciendo me parece todavía inimaginable, es más ciencia ficción que otra cosa. Antes de empezar con preguntas, me gustaría saber si alguien de aquí en Zoom quiere preguntar algo, que levante la mano y le damos paso, o voy leyendo las preguntas. Si hay algún tímido, que lo escriba en el chat, y si hay alguien que quiera preguntar directamente, español, inglés, lo que queráis, pues os doy un par de segundos. Y si no, empiezo a leer preguntas. De hecho, hay preguntas técnicas, hay preguntas sobre enfermedades de preeclampsia, o sea que vamos a dar un segundo. Bueno, mientras yo aprovecho y te hago un par de preguntas, que hoy curiosamente hemos tenido un seminario aquí de TENEX sobre Senium. No sé si conoces el nuevo sistema de TENEX. ¿Cómo crees que puede ayudar Senium a la hora de eliminar? Es que yo siempre pienso que la parte de la secuenciación es una de las partes que para nosotros es la más complicada por computación y por memoria RAM, que no sé vosotros, pero nosotros tenemos muchos problemas. Y a mí toda esta parte muy parecida a lo que hace NanoStream de obviar toda la parte de secuenciación, ¿tú crees que perderíamos esa información? ¿No?
Muchas gracias por tu pregunta. Ya no estoy compartiendo pantalla, ¿verdad? ¿Me ves?
No, se te ve perfectamente.
Sí, bueno, yo creo que son técnicas diferentes que depende de lo que queramos ver, será una cosa u otra. Te cuento un poco, nosotros también utilizamos Senium, bueno, hasta ahora hemos utilizado in situ sequencing que se vendía por Cartana y Senium es una versión mejorada de la técnica.
Sí.
Y básicamente lo que Spatial Transcriptomics te da unbiased information, pero no te da single cell resolution, que con bioinformática puedes tener más o menos una aproximación de qué células tienes, pero sigue sin ser single cell resolution y nunca, aunque disminuya la cantidad de células, nunca verás la boundary de la célula. Entonces, lo que te permite in situ sequencing es, bueno, pues tener información de la single cell resolution y definir a lo mejor niche de una forma mejor porque puedes ver las células que están alrededor. También al tener más resolución, lo malo de Visium es que si vas a la expresión de un particular gen, pierdes la expresión de alguna forma porque es como, bueno, tienes al final 3000 genes por spot que son los que van a estar más expresados, pero pierdes información. Entonces, con esto puedes generar información a más detalle. Por ejemplo, si te interesa mirar gradientes de un receptor o un ligando, esto es muy difícil de ver en Visium. Yeah. Lo malo es que, claro, son al final 100, 200 moléculas que puedes mirar. Sí. Pero bueno, de alguna forma tampoco lo que puedes hacer también es, bueno, primero seleccionas estos 200 genes, interrogando tu single cell, ya hay métodos para seleccionar los más informativos. Eso. Y luego puedes imputar con el single cell data. Así que yo creo que es complementario, sí.
Y mira, relacionada con eso hay una pregunta sobre StereoSeq. ¿Conoces la técnica?
Pues la verdad, sinceramente, no la he utilizado. O sea, sé que existe, o sea, simplemente es similar es para de convolve, pero la verdad, sinceramente, no podría opinar sobre ella porque no la he utilizado.
No, bueno, aquí nos hablan de que eliminas estos factores del spot, ya no tendrías entre 1 y 20 y tantas células. Al menos ellos, bueno, estos de BGI, ellos dicen que es realmente lo que sería célula. Entonces, no sé, me preguntan tu opinión, si puedes dar alguna, si no la has usado.
Pero sé que es también un método bioinformático, ¿cierto? Yo creo que al final te dará una probabilidad, así que me imagino. Yo sé que hubo un benchmark, pero te digo, hablo sin saber porque yo no... Hubo un benchmark y ponía que Cell to Location era de los que más accurate eran y por eso lo estamos utilizando. También porque, bueno, es de mi colega Omer Bayraktar, que participamos en el paper, pero no puedo opinar porque yo no he comparado los dos métodos, así que mejor no opino.
Vale, a ver, de parte técnica creo que ya más o menos he acabado. Mira, me preguntan por aquí que si has trabajado con muestras con preeclampsia, con el conocimiento y todas las aplicaciones clínicas para mejorar esta enfermedad o prevenir el desarrollo de preeclampsia.
Mira, no, la verdad es que no. Y también, bueno, es que el problema de trabajar con preeclampsia es que ya te vas a finales del pregnancy. Y el problema es que esta invasión, o sea, esto es un poco como el cáncer, ¿vale? Los elementos que ocurren al principio y que crean el ambiente para el cáncer, o en este caso para el buen desarrollo de la pregnancy pasan al principio, pero cuando miras al final lo que ves es más las consecuencias, no tanto las causas. Entonces es interesante, pero tiene un problema, que es que ves un montón de inflamación y ya no ves lo que está causando, porque ya no hay invasión solo ocurre al principio. Mucho lo que pasa en cáncer también, que por eso se utilizan muchos modelos de ratón, ¿no? En cáncer, porque quieres verlos, pero aquí como los modelos de ratón no sirven para ver la invasión, por eso tenemos que movernos hasta estos tissue banks y de aquí que la genómica sea tan interesante.
Perfecto. También nos pregunta cómo estudiáis las células gigantes, traducción, en un single cell, porque normalmente suelen ser multinucleadas.
Correcto.
Y para diferenciarlas de los dobles, vamos.
Correcto, y esto es por la razón por la que utilizamos single nuclei. No, perdona si no he ido mucho detalle.
Sí, lo has dicho.
Hemos utilizado single nuclei porque son multinucleadas, entonces a nivel de single nuclei los puedes capturar, pero no a nivel de single cell no los capturarías. Eso es así.
Perfecto. Creo que tengo a Alberto Sola levantando la mano. ¿Alberto?
Sí, hola, buenas. Rosario, enhorabuena por tu charla y muchas gracias. Yo te quería preguntar acerca de tu opinión sobre la integración de datos ómicos de single cell sequencing con las técnicas de imagen, como por ejemplo esto, no sé si Cosmic o algún tipo de estos de single cell imaging.
Bueno, nosotros, mi opinión, lo que he hecho es lo que nosotros utilizamos más de imaging, que ha sido in situ sequencing, que bueno, o sea, que no tengo familiaridad con Cosmic, pero debe ser parecido. Pero básicamente el concepto es el mismo. Lo que haces es, a nivel probabilístico, en realidad lo que haces es como utilizar los genes de alguna forma que son comunes para integrarlo, y lo puedes hacer, por ejemplo, modificando cell-to-location, porque al final es un framework de integración de datos. Entonces lo puedes hacer igualmente.
Vale, gracias.
Porque al final es un Bayesian method, entonces puedes cambiar los parámetros, pero sería lo mismo. Al final lo que hace es mirar los genes que están en común y utilizarlo eso para hacer la deconvolution o la integración. Entonces sería lo mismo para técnicas de imagen.
Tenemos una pregunta más en el chat. En relación al tratamiento informático de los datos, en el ámbito de single cell hay avance constante y una gran falta de consenso. Entonces quería preguntar tu opinión sobre cómo es mejor aproximarse a este tipo de análisis, si confiar en soluciones comerciales, artículos, pipelines, o cómo formarse para poder elaborar tus propias soluciones.
Estamos hablando de bioinformática, ¿verdad?
Sí, seguro.
Vale. Sí, a ver, yo creo que es complicado porque al final yo creo que no hay una solución que sea buena. Entonces, por ejemplo, esto especialmente cuando integras datasets que son de diferentes orígenes y tienes diferentes batch effects, o cuando comparas cosas que no son lo mismo, por ejemplo, in vitro, en vivo, como estaba diciendo. ¿Y qué pasa? Que muchos de los algoritmos, cuando tú integras juntos, lo que pasa es que te fuerzan la integración, entonces puedes tener cosas integradas y decir que es lo mismo cuando realmente no es lo mismo. Entonces, realmente creo que no hay una solución de decir, haz este pipeline y ya está. Creo que la idea es mirar los datos de muchos ángulos. ¿Y cómo puedes hacerlo? Pues como estaba diciendo, por una parte integrar en el sentido de integrar en el mismo manifold y ver qué poblaciones se van juntas. Luego también lo puedes mirar de otro ángulo, que es mirar markers, analizar siempre por separado y ver markers que son comunes cuando lo analizas de una forma y de otra. Finalmente también, como estaba diciendo, utilizar métodos como Machine Learning, hay otros en los que tú, si tienes dos datasets diferentes, lo entrenas en uno y luego lo proyectas en el otro. Entonces, básicamente lo que tú quieres ver cuando identificas una identidad celular es algo que es estable. y la única forma que hay de ver si es estable es probar diferentes pipelines, y no solo en términos de pipelines de convol de diferentes formas, pero sí mirar, como está diciendo, comparar los datasets de diferentes formas, como se está diciendo, integrar juntos, comparar datasets o mirar markers. Y solo cuando te coinciden es cuando estás confiada que lo que afirma es que una cell type se está integrando, realmente se está integrando. Y luego, claro, añadir diferentes layers te ayuda, por ejemplo, el spatial, porque ya tienes confianza que lo defines por single cell y también a nivel de spatial. O sea que mirar los datos de diferentes formas es key. Y lo que yo he presentado aquí es como un consenso, pero hay un montón de análisis que hemos hecho para estar seguros que esos son los clusters. De momento, no creo que haya otra forma.
Muchas gracias, Roser. Creo que no hay ninguna pregunta más. Te agradezco de nuevo enormemente la charla de parte de la sociedad, de parte de toda la gente que está conectada. Como te comentaba antes al principio, gente de background muy diferente, gente con ya mucha experiencia, gente con menos experiencia, y yo creo que sobre todo con lo que se quedan es con las ganas de poder trabajar en este campo y nada, y seguir avanzando en enfermedades, en el estudio y en el conocimiento. Te agradezco tu pregunta, tu charla, de contestar las preguntas y nada más. Te saludo y un abrazo muy grande.
Pues muchísimas gracias a vosotros por atenderme y también por darme la oportunidad de presentar. Ha sido un lujo estar aquí y escuchar vuestras preguntas. Gracias.
Gracias, hasta luego, Roser.